So I'm Heather Flanagan-Steet. I'm the Director of Functional Studies here at the Greenwood Genetic Center. My role here is multifold. One of the things I do is, as a trained developmental biologist who currently primarily works with zebrafish, one of my goals is to use the zebrafish to model rare genetic diseases, giving us a tool to gain a better understanding of how you go from a specific genetic change to a series of clinical features in our patients. The other thing that I do is, with the modern suite of diagnostic approaches that we have to help our patients receive a diagnosis for their clinical symptoms, one of the things that's happening is we are getting information about genetic sequence that we don't understand. So for example, often our patients, we find a genetic change in a particular gene for them, but if that change hasn't actually been associated with a known disorder or with a specific set of clinical features, it can be very difficult to evaluate whether that change is relevant for our patients. And we call these changes VUSs, or variants of uncertain significance. And one of my jobs as Director of Functional Studies is to help understand whether that change is actually pathogenic or problem-causing for our patients. And we can do that in cell systems as well as animal systems. But it can be very powerful to do those sorts of analyses in an organism like the zebrafish. So we've long used the zebrafish to study development, particularly because they develop outside the body, unlike a mouse or other mammals, which develops in utero. The fact that the zebrafish develops outside the body allows us to actually watch numerous events during early development that we would not normally be able to see. And for pediatric disorders, often the major changes that are affecting our patients are happening during that developmental period that a mammal would be in utero. So the fish is very powerful. It gives us the ability to look at those events and really ask questions as to how a particular genetic change led to alterations in tissue development. Another reason the fish is very powerful is they're very small and they're fairly transparent. So the ability to see into the animal's brain is much easier than it would be in the mouse. Or the ability to see in their bone and cartilage or their heart developing is much simpler than it would be with something like a mouse or a rat or even a cat or dog, which are also occasionally used to model human disorders. Fish surprisingly share a great deal of genetic homology with humans. They have as much as 70% homology across their genome. And if we actually just look at known disease-causing genes, that homology goes up to as much as 85 or 90%. So they're really a great model for human disorders in many cases. And their tissues actually develop very similarly to ours. And surprisingly, other than the lungs, they have pretty much every tissue that we have. So they can be used for structural disorders, bone and cartilage, heart defects, neurological defects. They can be used to study metabolic disorders. They have a liver, they have a pancreas, they have a very similar blood composition. And they also have a similar immune system. So there are a number of disorders that can be modeled in the fish. We have the benefit of numerous tools and technologies in the fish that assist these sort of analyses, including microscopic tools as well as biochemical and genetic tools. Okay, so I'd like to welcome you to the Allin Aquaculture facility here at the Greenwood Genetic Center. What I'm going to do today is take you on a tour of our facility, which is actually divided into three major areas. One that we call the life support room, one that we call the preparation room, and one that is the housing room where the animals actually live. So this facility is a system essentially that's a fully automated recirculating system. And what I mean by that is there is water flowing through all parts of the system at all times. So when we enter the housing facility, one of the things you'll see is that we have hundreds of tanks housing about 10 to 12 zebrafish per tank. Those tanks are all receiving water continuously, and water's flowing out of them continuously. So we actually create a current in our tanks, like what you would find in the rivers where these animals live. The life support room essentially is the power room that runs the entire facility. It has a series of large pumps that push water in a circle around the housing unit. So they push clean water from the life support room out into the housing room, and that's done through pipes that are drilled through the wall. So we've been able to actually isolate our life support room away from our housing room, and as you can tell, this is really wonderful for both us and the fish, since the pumps in this life support room are quite loud. So the other purpose of the life support room is it cleans the water that we are receiving back from the housing unit to provide a nice supply of low toxin, low bacteria water for our fish. This works partly by passing the water through a series of filtration units that clean all the sediment and debris out. That water then passes through a large tower that houses millions of small glass beads. These small glass beads actually have two different kinds of bacteria that grow on their surface. Those bacteria are actually what physically clean our water. So the fish are going to give off primarily ammonia, and the first bacteria on those beads is going to convert the ammonia to a nitrite for us. The second bacteria is going to use that nitrite that was given off as food and convert it to a nitrate. And the nitrate is going to be the least toxic of the materials to our animals, so we can then take that water and send it back to the system. We also have a large, what we call RO unit, or reverse osmosis, that takes water from the city, just simple tap water, and removes all the minerals and toxins and debris from that and holds it in a very large tank. And that allows us each day to push clean water into our system. We do about a 10% water change daily, and that helps reduce the nitrates that would be building up in the water, or any sort of other material that would be building up. Since we're pushing water back in that doesn't have salts and hasn't been pH balanced, we have a series of dosing units, one being a salt tower and the other being a carbonate tower, that allows us to put a little bit of salt and rebalance the pH. And this is done because a series of probes continually read the pH and salt in our system, and will tell those dosing towers when they need to put a little bit of carbonate or a little bit of salt to keep our water in a range that our fish like, that's healthy for them. The system also has a series of alarms that if anything falls out of those ranges, either the pH, the temperature, the salt, if a pressure gauge gets out of range, I receive a call on my phone that tells me someone needs to come in and look at this. So this is a really wonderful thing both for us and our fish. So when we leave the housing room, we have a second area called the preparation room, and that's what's here on my left. This room is essentially a kitchen where we clean all of our glassware and our fish tanks. It's where we raise food and make salt waters that we need for the animals' food. It's also where we manipulate those embryos genetically. So many of you have heard of CRISPRs and TALENs, or recombinant DNA technology. This is an area where we can introduce exogenous DNAs, or we can put a CRISPR or a TALEN into our animals and modify their genetic background. So one of the things that happens in the preparation room is we prepare food for the animals. We have to remember that this is a live organism whose life cycle involves chasing its prey and eating it a live diet. So one of the things we do to recapitulate as close to their natural habitat is we actually raise a live diet for them. One of the foods that we feed them are brine shrimp. Many of you may know them as sea monkeys. These are raised every single day in a harvesting cone, which has a salt level of ocean salt concentration. We warm these with a heater and we keep the organism bubbling. This allows them to hatch out of a shell. Tomorrow, when they've all hatched out of their shell, we'll harvest them out of this cone into a simple squirt bottle that can be used to feed the fish tanks one at a time. They eat this diet twice a day. So for lunch and dinner, they receive brine shrimp diet. In the morning, we actually feed them a complex flake that gives them all the vitamins and minerals they need. So if you're just raising fish at home, you don't need to feed them all of these nutrient-rich diets. But we're trying to get these animals to lay large numbers of eggs so that we can study developmental processes. And that takes feeding them these specialty diets that keeps them very healthy and robustly able to give us large numbers of embryos. So welcome to the housing room. This is the room that the animals actually live in. The room is composed of a series of 14 large metal racks. What you may not be able to see is that there's water coming in through the top of each of these racks and flowing down to each of these shelves, which have a series of tanks on them. Each tank has about 10 to 12 fish of the same genetic background. One of the things that you'll see is there are small blue hoses coming into the front of each tank. Those are bringing in the fresh water that we cleaned in that life support room. That water flows to the back of the tank where there's a small baffle that will push the dirty water out of the tank. So these animals are on a constant flow of clean water coming in and dirty water going out. That cleans their water and keeps their environment safe, but also creates a current like what you would find in a river, so that we're really mimicking the natural habitat of these animals. Each of these tanks has about 10 to 12 fish. Mostly we'll try to keep their sex ratios fairly similar, so somewhere between 5 and 6 females and 5 and 6 males in each of these tanks. The system can house several hundred tanks. We can house up to 10,000 fish, depending on the density in each of these tanks. We're currently running just over 0.5%, so we have about 6,000 fish right now that are divided into about 60 to 65 different lines of fish. So what I mean by a fish line is each of these 60 to 65 different groups have different genetic backgrounds. Some of them are fish carrying disease-specific mutations. Some of them are fish carrying what we call transgenic reporters. So some sort of reporter gene that we've put into these animals that allow us to study development. And typically these are fluorescent proteins, often marking specific tissues. So we have lines of fish, for example, that developing chondrocytes are labeled with GFP, so they glow green. And that's one of the tools that allows us to actually study that tissue developing in the context of a particular disorder. We have similar transgenic lines that label tissues in the heart. We have ones that label specific types of neurons if we're interested in studying an intellectual disorder or a neurological disorder. And then we have a number of animals that have specific disease-causing mutations that are allowing us to specifically understand why different tissues develop differently in the context of those diseases. Once we understand that, we can use those same animal lines to look for therapies. We can do drug screens, we can do other genetic manipulations, and ask whether we can improve tissue development for our patients. In addition to the adult housing component of this room, the room also has a nursery. So one of the things that we have to do to maintain all of these lines is we continually have to raise them. There's no way to easily freeze down animals, so we do have to continue to make new generations of each line. That's done in a nursery where small animals can grow before they go onto the system until they're large enough to go onto the system. So probably two of the most common questions people ask when they enter the facility is first, how do we tell a male from a female? So the body architecture of the male and female are slightly different. The female is typically a bit larger. She's going to have a bit larger belly. Her belly is going to have a little bit different coloring than the male. It's typical to be more white or silvery. The males tend to be more slender. They're often described as having a torpedo shape. And they tend to have a much yellower underbelly, and their lower fins tend to be much yellower. The other question that we get a lot is, how do we get eggs from these animals? So I think you're all well aware that typically if fish are going to lay eggs in the river, they could eat those eggs, or those eggs are going to fall down into the rocks. So fish like to lay their eggs in the shallows of the river where there's a lot of pebbles and rocks. So one of the things we do is to try to mimic that. So we've created what we call a chamber system. This is just a small Tupperware container that fits nicely down into the tanks. That we have a piece of black mesh with a series of holes, and then we can put plants in these. The plants provide both comfort for the animals, allowing them to swim and feel safe. They really want to lay their eggs where they feel safe and think the eggs will be protected. But it also has a physical component to them. As they're swimming through these plants in the water, they'll rub on the sides of the plants, which will stimulate the sides of their belly to drop eggs into the water. The male can then swim past those eggs and drop sperm, fertilizing them. Those eggs will then fall down in this chamber, which prevents the animals from getting to them and eating them. We can pull this chamber out of the water. We can remove this piece, and we can collect the eggs that fell down in it. If we want to continue to get eggs from them over the course of several hours, we can drop this chamber back into the water then. Some of the other questions we get are, do the animals lay eggs all day long? Is that something we have to be concerned with? And in fact, fish tend to lay eggs when the sun comes up. So one of the things that we do to mimic their natural habitat is they live in this facility on a light cycle. So they have 10 hours of darkness broken by 14 hours of light. And we have a cycle system that turns our lights on and off at the same time every single day. So these animals, after the lights come on at 8:30 in the morning, will start laying eggs, just as if the sun had come up in the wild. They will typically only lay eggs for a couple of hours, and then they'll be finished for the day. And most of our animals will not lay eggs at other times of the day. So that really allows us to prepare, drop these chambers into the tanks, and collect eggs at very regular times throughout the day. So Lynn has collected eggs using the chambers that we showed you earlier. She collected them simply into a petri dish, and now what she's doing is picking them up one at a time and placing them in what we refer to as an injection tray. And what this tray has is a series of small grooves that the eggs will fall down into, and allows us to inject large numbers of eggs very quickly. So she's doing something we called arraying the eggs, getting them all lined up in a nice neat row to pass over to Tong, who will then inject them using her microinjection setup and small glass needle. So one of the ways that we can use zebrafish is we can change gene expression in the animals. For example, if we know that a particular disorder is associated with loss of a particular gene's expression, we can remove that gene in our developing organisms. So Tong, a postdoc in the lab, is currently doing that, and what she is doing is she is using a microinjection set up, a very small needle that she can introduce into developing zebrafish embryos as an egg, and she can introduce either a new gene or she can introduce something that reduces expression of a particular gene, or she could introduce a CRISPR or TALEN. And in this way, creating a developmental model for a particular gene or mutation that we're trying to study. She can inject several hundred eggs in a single sitting. Typically, we inject anywhere from 200 to 1,000 eggs in a particular experimental window, usually within a couple of hours. And those animals can then be allowed to develop, and we can ask how altering that gene's expression impacts the development of the organism. So one of the questions that we often get when we're giving a tour of the facility is whether there's any governmental or organizational oversight when you're raising an organism like the zebrafish. And what surprises many people is that there is a tremendous amount of regulation and rules that we have to follow, and a tremendous amount of governmental oversight. So the GGC is a PHS accredited facility. What that means is the public health system of the government has accredited us, saying that we are able to use these organisms in biomedical research. It allows us to use NIH or government grant funds to do biomedical research. And really what it is saying that we adhere to a set of rules and regulations with regard to how we raise these animals, how we handle them, how we treat them, how we care for them, and even how we experimentally manipulate them, all falling within a set of parameters that minimizes either pain or distress to these animals. It ensures that we are not raising animals that we don't need, but that we keep our numbers within the limitations of what is required for our work. Okay, so this is the confocal microscope that we use to look deep into the embryo's tissues. The confocal is very useful because it actually allows us to get single molecule resolution relative to a more traditional microscope, which doesn't allow you to do that. It also allows us to look at a whole embryo or a whole animal and look deeper into that animal beyond the surface. So as we were talking about earlier, one of the great powers of the zebrafish is the fact that we can watch development happening outside the body. In order to do that, however, we need to be able to look deep into the animal, perhaps into their brain or deeper into the cells in their heart or the cells in their developing bones. And this instrument allows us to do that. We can get images of individual cells, or we can get three-dimensional images that we can reconstruct and rotate and get information from multiple angles. Here you're looking at embryos that have been stained with a dye called Allin blue that highlights their developing cartilage, and you can see that the wild type jaw that you're looking at looks very different than the jaw on the right, which is from one of our disease models for a disease called mucolipidosis 2. And one of the things you see right away is the disease jaw is much smaller. It's got a very different shape than the normal animal's jaw. And this provides the basis for us to start asking developmentally related questions as to why that jaw is smaller and shaped the way it is. In addition to these sort of histological stains that we can do, I had mentioned that there are a number of tools we have in our zebrafish lines, particularly these transgenic lines that label different tissues that are developing with fluorescent proteins. And so we have a number of different lines, one that labels motor neurons with GFP and one that labels chondrocytes. And particularly in this lower panel, one of the things you see are developing motor neurons. So what we're able to do with these is actually watch in real time as the cell of this motor neuron extends its axon out into the muscle. We can ask if it extends its axon in the same time as a normal scenario, if it takes a different path as a normal animal, particularly useful when we're studying neuromuscular disorders or disorders associated with progressive loss of motor function. We can use these sorts of tools to ask if that has anything to do with how their motor neurons developed. We have another transgenic line that I had mentioned that labels the chondrocytes and cartilage with GFP. And again, what that allows us to do is ask in a disorder like mucolipidosis 2, where there is a deformity of the cartilage, we can see why that cartilage is formed differently. In particular, in this case, one of the things that was clear is that while wild-type chondrocytes are sort of long and oval shape, the chondrocytes in the mucolipidosis 2 animal were small and round, and they were organized in a very different way. The other thing that we can use these animals for is to look at different proteins and how they're expressed. And one of the things you see in this picture is this very heavy red stain in these middle panels. That red stain is for collagen around those chondrocytes. And what you see is the wild-type animals are not expressing as much of that red collagen that the disease animals are. And that was a big clue to us as to what had gone wrong in the development of this tissue. But the point here to be made is that the power of the zebrafish is the ability to watch these things happening and allows us to form what the actual questions we should be asking are.